Good afternoon, student. As we have started a fifth unit from the last lecture, so today we are going to see the explant response, that is organogenesis. So what we are going to see in this topic, we will see the definition, the types, and factors affecting organogenesis. So first we will see what is meant by organogenesis. So first we will try to understand this word. So what is meant by genesis? That means formation. Formation of what? Organ. So that's why the term or the word is organogenesis. Now we will see definition. So you can define the organogenesis as the development of adventitious organs or primordia from the undifferentiated cell mass in tissue culture. So that's means callus, and that is done by the process of differentiation or the production of adventitious roots and shoots from the cells of the tissue culture, that is known as organogenesis. So in simple words, that means it's formation of organs. What kind of organs? That is root and shoot, and this is the result of differentiation. So that means the callus, from the callus there is formation of shoot and roots, and that is happens through the process of differentiation. So now we will see the types. So you can see the totipotent cell that will differentiate and form the organs. For example, callogenesis, rhizogenesis, and colorhizogenesis. So what is meant by collogenesis? That means it is the type of organogenesis by which only the adventitious shoot bud initiation takes place in the callus tissue. So that means collogenesis means formation of only shoot. So next is rhizogenesis. So rhizo, that is this word is related to roots. So it is the types of organogenesis by which only the adventitious root formation takes place in the callus tissue. So what is rhizogenesis? Only the formation of roots. Now what is meant by colorhizogenesis? So there is formation of shoot and root. So now you are going to tell me, so which type of explant that can be used for the colorhizogenesis. So that means you will get shoot and root at a time. There is no only shoot or root. Now we are going to see the factors affecting organogenesis or influencing the organogenesis. First is size of explant. The size of explant should be not too small or too large. The explant part should consist of parenchyma, vascular tissue, and cambium. So why? Because that contains the regenerative ability than the smaller explant. Then source of the explant. So keep in mind in plant tissue culture, the response is going to vary with the plant species. For example, the source of the explant, that is going to vary depending upon the plant species. For example, in case of Solanum, Nicotiana, and Begonia plant, the leaf and leaf fragments are the better explant. Then in case of Hylam plant, the bulb that is used as a explant. So next is seasonal variation. So you will find some plants gives best response in some specific season. For example, the bulb scales of Lilium plant, so that can give best response when collected during spring. But the same explant when collected in the summer or winter season does not produce any bubble. So this is one example of Lilium plant. So next is oxygen gradient. So there is formation of oxygen gradient. The air can pass through the closure. For example, the non-absorbent cotton that is used as a closure during the plant tissue culture. And what happens? So that regenerate the or that is generate the oxygen gradient. For example, for rooting requires high oxygen gradient, and for shoot formation the oxygen gradient is reduced. Next is quality and intensity of light. Generally in the plant tissue culture lab, the 16 hours per photo period is maintained. So we are going to see this factor with example. For example, you can see the Nicotiana plant. So when it is illuminated with high intensity of light, so that is 10,000 to 15,000 lux, so it can produce the shoot bud or embryo from the callus. And generally the blue region, so obviously this blue and red light region comes in visible region. So this blue region spectrum promotes the shoot formation, while the root light spectrum induces the rooting. Next is temperature. So most of the plant species require temperature, or the temperature which is set in the plant tissue culture lab, it is around 25 degrees Celsius. Means at this temperature the plants can grow, or that plant can give best response. Next is plant hormone. So we have seen in the plant hormone in the history of plant tissue culture lab. So the Skoog and Miller demonstrated that the different types of organogenesis can be achieved by varying the concentration of plant hormone. So what concentration you are going to vary? You are going to vary the concentration of auxin and cytokinin. When the cytokinin concentration is high, that means you are going to add the low concentration of auxin. And this high cytokinin concentration will induce the shoot formation, and low concentration of cytokinin will induce the root formation. So that means high concentration of auxin is required for root formation, and high concentration of cytokinin is required for shoot formation. Then this is important for you. So there will be question on types of organogenesis, that is direct, indirect. So we are going to see what is mean by that. And this type of organogenesis, it is depend upon the hormonal combination or the concentration of plant growth hormone. So this type of organogenesis that is depend upon the type of organ, but this type of organogenesis that is classified depending upon the hormonal combination. So what type of combination of auxin and cytokinin? So first we will see direct organogenesis. So in direct and indirect organogenesis, direct organogenesis is preferred. So why? That is we will see later. First, what is meant by direct organogenesis? So direct organogenesis is there is formation of organs directly from the explant. So that's mean the explant will develop into meristemoid. So there will be division of cells, or the explant will differentiate and form the meristematic cell, and that will give rise to organ, root or shoot. So you can see there is not callus phase. The explant will differentiate, and that differentiated cells will form the organ, that is root and shoot. So there is no callus phase, and that's why the direct organogenesis is preferred, because what happens in callus phase, there is undesired variation of clones happens. That's mean the genome becomes unstable, and there is chance of the change in the ploidy level. You will find the somaclonal variation in the plant which are regenerated from the plant tissue culture. So now we will see indirect organogenesis. So in this what happens, the explant will differentiate into callus, then the callus will form meristemoid, and then there will be formation of organs. So in this indirect organogenesis, there is callus phase is involved. So that's why it is called as indirect. So this is about organogenesis. And keep in mind the auxin and cytokinin ratio. When auxin to cytokinin ratio is high, so there will be formation of fruit. So what is meant by auxin to cytokinin ratio is high? Means there is high concentration of auxin and less concentration of cytokinin. And when there is auxin to cytokinin ratio is low, that's mean concentration of auxin is low and cytokinin is high, and then there is formation of shoot. Okay. Thank you.